Good morning and welcome everybody for today's webinar entitled Xenografts and Allografts Allied in Our War on Cancer. I'd like to briefly go over some of the technical pieces before we get started on the webinar. A copy of the presentation will be available right after the webinar. If you have any kind of technical difficulties, please raise your hand in the control panel and our technical staff will assist you. To ask a question during the webinar, please use the Q&A tab in the control panel and send your questions to our panelists. At any point in the presentation, if you want to get back to the control panel, click on the blue button on the floating top panel, and I'll get you to the control panel. Now, with that, let me start introducing our speaker for today. It's Dr. Dave Grass. Dr. Dave Grass received his doctorate from Columbia University in molecular virology. He did his postdoctoral training at the Sloan Kettering Institute for Cancer, working with Dr. Elizabeth Lacy in molecular immunology. He joined Taconic, formerly DNX Biosciences, in 1992. As a senior scientist, he was responsible for establishing a transgenic animal model generation service. He he supervised research aimed at generation of rodent models for inflammatory conditions and human lipoprotein metabolism. Over the past decade, he has authored and co-authored several peer-reviewed publications. He was instrumental in creating the phenotyping platform used for characterizing more than 200 knockout lines and over 400 compounds. He is currently our VP of Business Development. So without further ado, I turn the floor over to Dr. Grass. Dave?
Thanks, Lex. And I want to thank everyone for taking the time to attend the webinar today. Hopefully we'll give you some information that's useful. We're going to talk about xenografts and allografts today. And in general, I want to talk just a little bit about Taconic and what and the approaches that we're trying to take to provide more predictive preclinical data. And basically what we're trying to do is to provide animal access, that's traditional strains, genetically modified animals, and also the ability to create new genetically modified animals. CMGS stands for Custom Model Generation Solutions. The ability to grow the cohorts of those animals at a defined health status for subsequent studies that either our clients can do or we can do. So we can provide phenotyping data on those animals that we've created or that have been created elsewhere, and we can also use those animals to do compound profiling, and that includes not only the traditional models but the innovative GEMs. Again, the idea here is to bring all these things together to create more predictive preclinical data. In oncology, we're taking an approach where we've brought in a number of different collaborators, and we're trying to bring a number of tools and services together under the platform that we're calling Onco. One of our partners in this is Onco Design, that brings a lot to the table for us. CIEA gives us access to a super immunodeficient animal, the NOG mouse. The Taconic cancer models, and we're going to talk about those today, are genetically modified animals that we've in-licensed. We have the rights to do studies in those animals. We have the rights to the Onco mouse license. And in general, we're trying to bring together as many different, all the different types of tools so that we can go from the high throughput in vivo screen, and we're going to talk about that a little bit today, the basic xenograft studies, and today we'll really focus on the orthotopic metastatic studies. And really, our platform gets to the more advanced translational in vivo models like patient-derived tumor models, advanced imaging technologies, and some of these things are being brought to the table by Onco Design so that we can look at drug-target interaction, mechanism of action, drug resistance, disease-associated biomarkers and even do these studies in mice that have reconstituted immune systems. Today, the focus will be really on these two boxes, the orthotopic metastatic models as well as the syngeneic allograft models and spontaneous tumor models. And first, I'll talk about the xenograft models, but I want to show really how we think of this. And if I go back to the previous slide, you can see that we look at these different capabilities and services that we can do and the animals that you use for them as a continuum in terms of being able to do high throughput in vivo screens and then really being able to move towards data that may be more translational and more sophisticated. Xenograft models, again, we think of as high throughput in vivo screens. One of the things that makes them really valuable and efficient is the ability to do the bioluminescent imaging, and I'll show you examples of that, which allows us to run these models as orthotopic models and not just sub-Q models. And that means that we can implant the tumors at the tissue where the tumor arose, so that we can take advantage of the lymph system and the circulating system so that the metastatic events that we get are similar to what you see in the clinic. And these types of studies are useful not only for looking at the primary tumor, but also for testing anti-metastatic therapies. Now, the spontaneous tumor allograft models are different in the sense that they carry specific defined mutations in relevant tissues. They're genetically modified. The mice are genetically modified so that you get spontaneous tumors. They acquire additional mutations which reflect tumorigenesis in humans as the tumors develop. And the tumors grow with intact microenvironment and retain the relevant tumor cell stroma interactions that you would get in humans as well. The models use immunocompetent wild-type mice. So basically, when I say the models use immunocompetent mice, that's if we're using them, if we're creating allograft models there. And they are useful for testing immunomodulating therapies, and they model and they resemble key aspects of human disease. So I will talk about the xenograft models first, and then we'll transition into the allograft models. The xenograft models take advantage of our ability to use this bioluminescent imaging technology. We like using this because we could create orthotopic models which better represent human cancer with respect to both the primary tumor location and the pattern of metastases. The challenges here are basically being able to do the surgery, so you need highly skilled technical staff to be able to do this. We're fortunate that we have people on our staff who are able to do this and have a lot of experience doing it. And again, the imaging technology really makes this possible because we're able to implant tumors in tissues and in places in the animal where you wouldn't be able to physically palpate and see how big the tumor is. For these xenograft models, we have several different types of cancer that we cover: breast cancer, ovarian, prostate, lung, colorectal, brain, melanoma, pancreatic. We can also create cell lines customized so we can take cell lines, genetically modify them so that they express luciferase and therefore can produce light. The difference between standard BioWare and BioWare Ultra is just the version of luciferase. Luke2 has been genetically modified is codon optimized for expression in mammalian cells. However, we get very good sensitivity with the Luke1 cell lines as well. We have a lot of experience working with them. So I'll talk about some of the orthotopic tumor models with spontaneous metastases. And here, there are 3 examples on this slide. One is our PC3M luciferase prostate, MDA231 luciferase breast tumor, and SCOV3 luciferase ovarian tumor. And you can see that in all the cases, after the tumor cells are implanted, either in the prostate, the mammary fat pad, or the ovary, you can see that the tumor grows over time in all the cases. One of the advantages of this imaging technology is that it allows us to see information that you might not always see if you were just using caliper measurements. So in this particular case on this breast cancer, you can see that I should mention that we're looking at a false color image where red represents the highest density of photons coming from the animal and blue represents the lowest density. You can see that on this animal, this representative animal, week 5, with the breast cancer cell line, you can see that we have an area of lighter intensity, so blue in the middle of this tumor, indicating that what we may be looking at here is a necrotic core, so that the tumor, which you might not see if you were just palpating or doing caliper measurements. Now, one of the things that we can do is if we cover the primary tumor with an opaque material, we can start seeing light coming from secondary tumors or distant metastases. And you can see that with the prostate model, PC3M, MDA231 breast, as well as the SCOV3, we are seeing tumors that are actually above the region of the primary tumor, indicating that there were metastatic events. Now, this slide shows, with the ovarian cancer cell line, shows that if you can do ex vivo imaging. So at the end of the study, we've taken out the tissues. These are the grayscale pictures on the bottom here. And you can see that if you look for the bioluminescence on the top, that there are tumor cells in the brain, in the lungs, in the liver, in the lymph node of the GI, in the bones, as well as in the muscle here. The ex vivo imaging, you have to be careful to do it quickly in order to see light from the tissue still has to be alive, so you have to work quickly and you have to do it consistently over each mouse, and we have a lot of experience doing that. Now I'll show you a couple of different examples. One is the breast cancer, where we can use compounds and basically show that this model will reflect what happens in the clinic. And the compound that we're using here is Taxotere. And the data we're looking at is showing that the tumors by caliper measurements as well as by bioluminescent imaging. And you can see that vehicle treated by caliper measurement and by imaging, those tumors continue to grow. The animals that are treated with Taxotere, you see a decrease in the size of the tumor by caliper measurements as well as by bioluminescent imaging. If you look at the metastatic growth here, we're looking at docetaxel in this example. You can see that the vehicle treated, there's an increase in, now we're looking at light, where we can see an increase in photons per second, but not in the animals treated with docetaxel. If you look at just the fold change, you again see an increase in the vehicle treated as opposed to the animals treated with docetaxel, where there's not an increase. So it is able to block the growth of the metastatic tumors. So if we look at, and we can quantify this, and we're looking at bioluminescence in the chest area, on day 1, 3, 6, 10, and 13 post-treatment. And you can see that initially, let me just focus on the furthest day out, day 13. You can see that there's a significant difference. The animals treated with docetaxel have not had their an increase in light over the time. So if you just look from day 1 to day 13, however, there has been an increase at day 3, day 6, day 10, and day 13 in the vehicle treated with respect to, and this again is just focused on the chest area metastatic events. We can do, again with this model, quantitative ex vivo analysis. We're looking here at the lungs and the lymph node. And just to give you an idea, you can see that almost all the animals have had metastatic events in the lungs. I think this is the one exception here. Many of the animals had it in the lymph nodes as well. And that's reflected in the data quantified here. So we're looking at the lungs. You can see that there's an increase in light in all of the animals compared to the animals treated with docetaxel. In the lymph node, you can see that some animals didn't have metastatic events, but a majority of them did when untreated. And all of the animals treated with docetaxel we did not see light coming from the lungs or the lymph nodes. In this case, the lymph nodes. One other example that I'll show is the PC3M, the prostate tumor model. In this particular slide, we're showing vehicle treated. The tumor continues to grow, and that's reflected by this representative animal here. You can see that from day 14 to day 36, the tumor continues to grow here. The animal treated with Taxotere, you can see that the actual tumor regresses. And if we look at, and this on the right side here really just shows the in vivo, the difference between the growth of the primary tumor and the metastases. And it's several orders of magnitude different, which is why in order to really see the light from the metastatic events here, you have to cover, you have to block the light coming from the primary tumor. Again, we're looking at ex vivo bioluminescence in the PC3M model, lungs, liver, lymph node of the GI, and you can see that we can quantify and show that the animals that were treated with the compound tend to have fewer metastatic events than the animals that were vehicle treated. And that's reflected, the tissues ex vivo taken out. You can see the vehicle-treated lungs, very bright compared to the docetaxel-treated ones. You can see the liver compared to the drug-treated animals, and also the lymph node. So this really summarizes the xenograft models that we have available. You can see that we've done sub-Q for all of them, but we've also done orthotopic and been able to see spontaneous metastases for many different ones. And we do have the capabilities of doing experimental metastatic models. That's either IV injection or intracardiac injection as well. This slide just shows that there are similar patterns of metastases in the models that we've run that I was showing you compared to human. And basically, what we're looking at is the percent of patients here, this is human patients who have metastatic events in the lymph nodes, in the lung, in the liver, in the bone, and so on. And red asterisk shows where we've detected metastatic events in the mouse models that we've run. So you see that we've gotten most of the major metastatic sites that you see in human, we're also seeing in our model. And that's true for the breast cancer model as well as the prostate cancer model that we showed. So I'll switch gears now and talk about the spontaneous and allograft models that we have. And these models are generated by creating genetically modified animals that have oncogenic mutations, specific mutations in relevant tissues. These models resemble key aspects of human disease, pathology, histology, and some of the molecular hallmarks. And a lot of work is still being done, but they do respond similarly to some of the therapies used in the clinic. And here are some of the models that we're working on now. We have breast cancer, lung cancer, colon available. We're in discussion for pancreas and prostate as well. We've in-licensed these models, I should mention. And the utility is that, again, they resemble tissue-specific mutations that are found in humans. And the tumorigenesis resembles what you see in humans. They have an intact microenvironment, so the interactions between the tumor cells and the stroma are what you would see in a normal tumor. They grow with an intact immune system, and they can propagate it easily and harvest it for molecular analysis. And one thing that I really do want to focus on is that cohorts can be generated easily for compound testing. And what we try to do for compound testing is basically create allograft models so that we're staging the tumors so that we have groups of animals that have tumors that are starting the study at the same size. So basically, we've isolated several different we've isolated tumors from, so far we've done this for the breast cancer line, and we have several isolates of tumors that we can run allograft models in. And this is just an example of the kind of study that we can do, where we can have a group vehicle treated, PARP inhibitor, and we can look at the effect of combining those two with cisplatin and then cisplatin with the PARP inhibitor. And you can see some data with the PARP inhibitor on the next slide. We're showing treatment for 28 days with a PARP inhibitor and 100 days, and you can see that compared to the vehicle control, where you're looking here at the relative tumor volume, you can see that the untreated control, that tumor volume continues to grow, but the inhibitor actually inhibits the growth. In this case, the animals were treated for 28 days, and then you can see that the tumors start growing after that point in time. In this case, they're growing for 100 days. You can see some tumors actually start being resistant to the treatment, but some of them continue to be actually not grow until later on. So these are the kind of studies you can do, and you can actually do the combination as well. This just shows survival with those different groups. Not surprisingly, the untreated group has the worst survival. The actual group that was treated with cisplatin and the PARP inhibitor actually has the best survival in this study. And you can see that the different treatments, this is another cisplatin and the PARP inhibitor for 28 days. This is cisplatin for 100 and PARP inhibitor for 100 days, and you can see that those are the two best groups. This data was published in PNAS by Rottenberg et al., and that work was done in the lab of Jos Jonkers at the Netherlands Cancer Institute. So we have isolated from the BRCA1 P53 tumor that we licensed in from that lab. We have several different isolates of tumor isolates that we can use for allograft studies, and that's the approach that we've try to take to more industrialize these kinds of studies. Again, this is a growing program, and really where we are is that we have triple negative breast cancer, this BRCA1 and triple negative. We also have invasive lobular carcinoma. We have non-small cell lung cancer and small cell lung cancer. In addition, we have the PERK-RAT model, and we're in discussions in terms of a pancreas and a prostate model that we can create allograft models from. So just to summarize, I've talked about xenograft models as well as allograft models. Xenografts are high throughput in vivo screens. We can use in vivo imaging. However, they're done you use immunodeficient animals for those. The allograft models are genetically defined in the sense that we know what mutations were first genetically modified into them. You use immunocompetent animals for these kinds of tests, and that allows you to test for immunomodulating therapies. And these models do resemble key aspects of human disease. So I want to thank everybody for their time and for participating in this WebEx, and I'd be happy to try to answer any questions that you might have.
Thank you very much, Dave. Just as a reminder to our audience, if you would like to ask a question, you can click on either the chat panel or the Q&A panel, and you can send our questions to our panelists. I have a couple of questions here, Dave. What type of mice is used in the SKOV3 LUC model, which is the ovarian cancer model?
I think that they're skid beige. I'll have to check for sure, but I think that they're skid beige mice.
Okay. Another question with the PC3 LUC model, do you need to sacrifice the animals for each of the time points to get the data?
No, actually. The nice thing about the imaging technology is that you can follow the animals over time, over several different time points, non-invasively. So that's generally how we do that. We do imaging at different times, and therefore you don't have a group that you have to sacrifice at each time point.
Another question here. You talked about the tumor cell stroma interactions in an allograft model. Can you explain a bit detail about it? Are you talking about signaling molecules here?
We're talking about the interaction between the tumor cells and the tissue around those tumor cells. And the nice thing about the allograft models and the spontaneous models is that you're going into the exact same strain, and the interaction is more similar to what you would see in the clinic. Not as much so with the human models going into the mouse, but it's as close as we can come. I think you tend to see, in all cases when you're using human cells, you tend to see the stroma, even when you do patient-derived tumors, you tend to see that the human stroma that originally you're implanting is replaced by mouse stroma. So there is an interaction there between the mouse cells and the tumor, and in the allograft model, that is probably as close as you can come to being similar to what you see in human, because you're not changing species.
Another one here. In humans, there are mutations that occur later in a tumor after a tumor has established. What type of model would you recommend to study those?
I think that the allograft models would be good to study those, as well as patient-derived tumor models that you can also access through Taconic. Both of those basically, the allograft models, we start with a defined genetic mutation, but they will pick up other mutations. We're still, as the tumors develop, and these are still being characterized, but for the patient-derived tumors, I think that those also, since they're derived from a patient, those mutations that you're going to see are, straight from the clinic, basically.
Another one here. Do you primarily see metastases in the orthotopic model rather than in the Xenograft model, or do you see similar levels of metastases in both? I'm thinking that they're talking about the sub-Q versus the orthotopic there.
Yeah, we tend to see metastases in the orthotopic models, and we think that it's because they are the cells are implanted and basically the lymph system and the blood circulation around those cells and around the tissue of where the tumor originated is similar to what you see in the clinic, which is not necessarily the case in the sub-Q tumors. So that's normally we do see it more in the orthotopic.
Thanks, Dave. A couple more questions here. You mentioned orthotopic models are good for studying metastases compared to the sub-Q model. How much do lymph nodes play a role in the metastases, or physical barriers like diaphragm and stuff pose a challenge to metastases?
Yeah, I mean, we think, as I said in in response to the last question, we think that the lymph circulation as well as the blood circulation is really key in terms of these metastatic events. And actually, the physical barriers such as the diaphragm often, gives us confidence, basically. So in other words, if we're setting up a prostate cancer model, any cells that are above the diaphragm aren't there, cannot be there just because they escaped when we were injecting, when we were basically implanting the cells into the prostate. They had to go there through the blood, through the circulation and the lymph. And so the barrier actually gives us confidence that what we're seeing is not really the product of leakage, but it's really a metastatic event.
Another one here. In your talk, you mentioned a lot about bioluminescence imaging for orthotopic models. Can one also use fluorescent markers? Is there any advantage of one over the other?
I would say that you can use fluorescent markers. It's much more difficult to do that because, at least with this imaging technology, with fluorescence, you require excitation wavelengths to get into the source of the light, or the fluorescent molecule, which is internal in the animal, and then the light has to shine out, and so there's scatter of light in and scatter of light out, and the signal-to-noise ratio for fluorescence in our hands is not as good as bioluminescence, which doesn't require any excitation wavelength. So we've been able to do some fluorescence imaging, and it works well with tumors that are close to the surface, or if you're imaging a structure that's close to the surface of the animal, but bioluminescence gives you more sensitivity.
Cool. Thank you very much, Dave. I think that's probably all the questions that we have for today. So we would like to conclude the webinar, and I would like to thank everybody for attending our webinar today. And please stay tuned for the future webinars that you can view them at www.taconic.com/webinars. And we'd like to invite you to the next upcoming webinar, which is on the 20th of June, entitled Antibody Pharmacology by Dr. Jean-Francois, who is the Technology Director at Onco Design. Again, I'd like to take the time to thank everybody for attending today's webinar, and hope to see you in our future webinar sessions. Thank you, and have a nice day.